Good afternoon, everyone. Today we will discuss about the green algae on the topic Oedogonium, salient features, occurrence, thallus structure and reproduction. Taxonomic position of Oedogonium, subdivision Chlorophytina, class Chlorophyceae, order Oedogoniaceae, family Oedogoniaceae and genus Oedogonium. Salient features of Oedogonium. The plant body is green, multicellular, unbranched and filamentous. The mature filament is differentiated at the apex and face. The reticulate chloroplast is present in each cell with many pyrenoids. In the division cells, caps are present. The cell division in the vegetative cells is much more elaborate. The vegetative reproduction occurs through the formation of A kinase. Asexual reproduction takes place in the multiflagellate zoospore. In each zoospore, flagella are arranged around the colorless beak-like region. Sexual reproduction is the advanced oogamous type. Based on the distribution of the sex organs, the plant are divided into two groups, the Machnandus and the Nonnandus forms. The Machnandus types, the antherial developed in the filament of normal size, but the Nonnandus type, the antherial growth on the small and thin male filament, top male or anandrium. Occurrence of the Oedogonium. The Oedogonium is a genus about 400 species. It is exclusively freshwater in habit and mainly occur in permanent and temporary freshwater body like ponds, lakes and swamp tanks. The filaments are attached to various substrate underwater or epiphytic or aquatic plants and other algae. A few species are terrestrial, growing on the moist soil. Thallus structure of Oedogonium. The plant body is Oedogonium is multicellular filamentous and green in color. The filaments are unbranched and uniseriate and arranged in a single row or attached end to end. The mature filament are differentiated into three types of cell. The lowermost basal cells are holdfast, the intercalary cells and uppermost apical cells. This diagram shows thallus structure of Oedogonium. Next, the basal cells are holdfast. This diagram shows basal cells holdfast. The lowermost cell of the filament is the basal cell. It is modified to form holdfast and heptatron. The holdfast is mostly found in aquatic species and it rarely occurs in terrestrial forms. The terrestrial species, it may produce rhizoids like outgrowths. The holdfast is long, gradually narrow and towards the basal parts. It expands to form a simple disc-like multi-lobe, a finger-shaped outgrowth or projection. The basal cells are usually colorless and do not take part in photosynthesis due to chloroplast being absent or poorly developed. It helps in the attachment of the filament to the substratum. Intercalary cells. All the cells of filaments lying between the apical cell and the basal cell are intercalary cell. These cells are similar in structure and long than their breadth. Some cells contains a ring-like structure in their upper part called a cap or apical cap. Such cells are called cap cells. The number of caps are a cell shows how many times it has been divided. The apical cells, the terminal cells are tarsal distal cell of the filament is called apical cell. It is usually rounded or acuminate towards the apex. In some species, the apical cells prolongs into a hair-like structure. The apical cell is green in color due to the presence of chloroplast and take part in photosynthesis. The cell structure of Oedogonium, the vegetative cells are the elongated and cylindrical with the slightly swollen or inflated upper portion. The cells are surrounded by a thick, rough and rigid cell wall. The cell wall is composed of three concentric layers, the outer chitinous layer and the middle layer pectin and the inner layer cellulose. However, the electron microscopy revealed that there was no cellulose layer. The mucilage is absent on the surface of the filament. The thin plasma membrane encloses the protoplasm. The protoplasm contains cytoplasm, the central vacuoles, reticulate, chloroplast and the nucleus. A large vacuoles is present in the center of the cell which remaining filled with the cell cap. The cell shape consists of excretions and secretions and inorganic compound. The chloroplast is a single large sheet like reticulate with the remaining embedded in the cytoplasm. It extends from one end of the cell to the other and completely encircles of the protoplast. The stand of the chloroplast are broad or narrow and are parallel to the long axis of the cell. Many pyridinoids are present in the chloroplast, particularly in the region of intersection. The pyridinoids are surrounded by a sheet of starch granules. There is a single large biconvex nucleus that lies near the middle of the cell. Internal to the chloroplast, the nucleus contains one or more nuclei and thread-like or elongated chromosomes. This slide shows reproduction in Oedogonium. The Oedogonium reproduces in all three methods: vegetative, asexual and sexual methods. The vegetative reproduction takes place. Oedogonium reproduces vegetatively through the fragmentation. The fragmentation Oedogonium filament can break into a small fragments. The broken fragments, which are detached from the thallus, they develop into a new filaments under favorable condition. The accidental breakage of filaments, drying or dehydration of the intercalary cells. The disintegration of intercalary cells due to conversion of zoospore or gametes. Change in the environmental condition. This is asexual reproduction. Asexual reproduction in Oedogonium takes place by the formation of various types of asexual spores such as a zoospore, abalonospore and acinides. The zoospore formation most common method are reproduction in aquatic algae. The zoospore are forming singly within a specialized cells. They called zoosporangium. Usually, a recently formed capsule functions as the zoosporangium. Several factors that control zoospore formation, including the high pH and carbon dioxide concentration of the medium, as well as the during diurnal rhythm of high light and darkness are significant. During the favorable condition, the zoospore formation beginning in a newly formed capsule of a filament. The zoosporangium heads filled with abundant reserved food material. The entire protoplast of the zoosporangium contrasts from the cell wall and becomes a rounded mass or oval shaped structure. The nucleus moves towards one end of the protoplast. This slide shows the formation of zoospore in Oedogonium. In the central vacuole disappear the chloroplast. frees itself from one side of the cell and becomes conical. The colorless semicircular hyaline area appears on the side close of the nucleus. This hyaline balding area later form the anterior region of the zoospore. At the face of the hyaline area, a single or double row plast granules or basal granules develops. The epiperous plast granules are connected by fibrous strand to form a complete circular ring. The latter and the single flagellum arise from each epiperous plast granules. In this way, a ground of about 30 flagella is formed around the face of the colorless peak-like area of the protoplast. This is when the zoospore is matured, as well as the zoosporangium split transversely near the apical cap region, and the upper portion of the cell is lifted off like a lid. During the liberation, the mucilate substance is secreted at the face of the zoospore, which helps in the liberation of the zoospore. The zoospore remaining enclosed in a delicate mucilaginous vessels for 3 to 10 minutes. The mucilaginous vessels soon disappears and the zoospore are liberated in the water. The zoospore is deep green oval, spherical or pear-shaped structure. It contains a single nucleus, an eye spot, a chloroplast and numerous vacuoles. The subapical ring of flagella is present at the face of the colorless peak-like anterior end, such as a flagellar arrangement is called streptocon. This slide shows germination of zoospore. After liberation, the zoospore swims up about an hour or more. Then it settles down and attaches itself to the substratum by its anterior end. After attachment, the flagella are withdrawn and the zoospore beginning to elongate. The lower hyaline part elongates to form a holdfast, and the upper part divided repeatedly by transverse division, forming a new filament of Oedogonium. A-kinase are formed during the unfavorable condition. In some species of Oedogonium, there are swollen thick wall, reddish brown, rounded or oval structure. A-kinase are formed in chain and are rich in starch or a reserved food material and in reddish orange oil. Under favorable condition, A-kinase germinate into new filaments by allospore. In unfavorable condition, insist a zoospore. Albinospores are formed in some species. Albinospores are thin-walled, non-flagellate, non-motile spore that are slightly oblong or spherical. The albinospore comes out to the parent cells and form a new plant with the onset of unfavorable environmental conditions. Sexual reproduction. In Oedogonium, the sexual reproduction is an advanced oogamous type. The sexual reproduction is more frequent in stagnant water than the running water. The alkaline medium deficiency and nutrients, light and dark period and increased temperature are influencing sexual reproduction. The gametes are produced in highly specialized cells called gametophagia. The male gametes or androzoids are produced in antheridium, and the female gametes or egg is produced in oogamia. That is female gametophagium. The male gametes and the female gamete are structurally and physically different. Distribution of sex organs depends on the distribution of sex organs. Oedogonium Species are divided into two groups, Macronandus species and Nonandus species. The Macronandus species, in the Macronandus species or Anthridium is produced on the filament of normal size, and the Macronandus species are of two types, monoecious types or dioecious types. Kynandospore types. In the Kynandosporous, Nonandus type are Endosporangia and Oedogonia are formed on the same filament. For example, Oedogonium concatenatum Ediandospore type in the Oedogonium. Ediandospore, Nonandospores types, the Endosporangia and Oedogonia are formed on different filaments. For example, Oedogonium ceticeratum and Oedogonium iengari, Anthridium and Macronandus species. The structure and development of Anthridium and Oedogonium are similar in both monoecious and dioecious type Macronandus species. In Macronandus species, Anthridium is developed in either the terminal or intercalary cell of the normal size filament. The initial cells that gives rise to Anthridium is called the Anthridial mother cells. Any type of cells, the vegetative filaments may function on Anthridial mother cells. The Anthridial mother cells divides transversely into two unequal cells, the upper small Anthridium and the lower large sister cells. The sister cells then divides transversely to form uniseriate rows of 2 to 40 Anthridia. Rarely Anthridia are produced singly. A structure and development of Anthridium and Oedogonium are similar in both monoecious and dioecious type of Macronandus species. This slide shows Oedogonium filament bearing Anthridia in the chain. In Macronandus species, the Anthridium is developed in either the terminal or intercalary cell, the normal size filament. The initial cell that gives rise to Anthridium is called Anthridial mother cells. Any cap cell of the vegetative filament may function as an Anthridial mother cell, and the Anthridial mother cell divides transversely into two unequal cells, the upper small Anthridium and the lower sister cells. The sister cells divides transversely to form uniseriate rows of 2 to 40 Anthridia. Rarely Anthridia are produced singly. This slide shows Oedogonium filament bearing Anthridia in chain. The Anthridium is a unnucleate, broad, flat, short cylindrical cells. The nucleus of each Anthridium is divided vertically by transverse or vertical wall into haploid nuclei. Each nucleus gets surrounded by some cytoplasm content of becomes pear shape. It develops a ring of flagella around a colorless portion, and it is one end and metamorphoses in a anthrozoids or sperm. Thus, two anthrozoids are produced in each Anthridium. The two anthrozoids are positioned side by side or one above to other. The lie in the Anthridium. The anthrozoids are spermatozoids or sperm are unicellular, uninucleate, multiflagella, oval or spear shape and yellowish in color. Morphologically, they are similar to zoospore but are relatively small in size and have few flagella. This slide shows Anthridium and liberation of anthrozoids. The liberation of anthrozoids from Anthridium is similar to zoospore formation during the asexual process. The wall of the Anthridium ruptures transversely and two anthrozoids are free into thin hyaline vessels. Soon as the vesicles dissolve and two anthrozoids swim freely in the water. The ribulated anthrozoids swim freely in water before they reaches the Oogonia and participate in the fertilization. This is Anthrium in Nonanthus species. In Nonanthus species, the Anthrium is developed on a short or top male flamen are called the top male or Nonanthrium. The top male flamen is produced by a germinate of a peculiar type of spore called androspore. The androspore are produced singly within the androsporangium. The androsporangia are more or less similar and appearance to the Anthridia or Macnanthus species and are produced in a similar manner from the mother cell by unequal division. The androsporangia are flat discoid cells that are slightly larger than the Anthridia or Macnanthus species. The protoplast of each androsporangium metamorphoses into a single androspore, just as in a case of the zoospore. The androspore is a unicellular, uninucleate, motile structure that has a subpolar growth of a flagella. It is larger than the anthrozoids but smaller than the zoospore. This slide shows Nonanthus species showing top male with Oedogonium. During the liberation, the androspore is surrounded by a mucilaginous vesicle which help in the liberation of the androspore. The mucilaginous vesicles soon disappears and the androspore are liberated into the water. After swimming about some time, the androspore settle down on the oogonial wall. The androspore germinate in a top male flamen or Nonanthrium. The Nonanthrium or top male can be few cells long. The lower rhizoids like elongated cell of a top male. It is known as the stalk. The stalk cell cut off one or more cells. It is stiff and form Anthridia. The protoplast of each Anthridia cell divided to form two sperm or anthrozoids, which are similar to a anthrozoids of a Macnanthus species. Each anthrozoid is uninucleate, unicellular, oval or pear-shaped and has a subapical ring of short flagella at the face of a colorless peak-like anterior end. The antheridae are released by the separation of a cap-like lid at the top of by the disorganization of the antherial cells. This is Oedogonium. In Oedogonium, the Oedogonium is a highly differentiated female chematogenum. It is generally intercalary but sometimes can be terminal. For example, Oedogonium palaiensis. The structure and development of Oedogonium are similar in both Macnanthus and Nonanthus species like Anthridium. Any actively growing capsule of the vegetative filaments may function as the Oedogonial mother cells. The Oedogonial mother cells divided transversely into two unequal cells, the upper large cells and the lower small size cells. This slide shows Oedogonium associated with the top male. The Oedogonium associated with the top male, the upper cells is rich in cytoplasm and the form of Oedogonium. The lower cells has a smaller nucleus than the upper cell and function as a supporting cell, supplotary cells. The supporters elongate conical cells. The lower cells may again function as a conical mother cells and undergoes similar division in repeated sequence to form 2 or 3 or more Oedogonial with a lower supporting cells. In some species of Oedogonium, the oogonial mother cells directly develops the oogonium. Supporting cell is absent in Oedogonium americanum. The oogonium is an oval, spherical or elliptical structure. It enlarges prominently and it is filled with resolved food material. The entire protoplast in an oogonium metamorphoses in the single non-motile egg or ovum or zoospore. The egg is green in color due to the presence of chlorophyll. This slide shows oogonium matured oogonium zygote within the oogonium. Initially, the zoospore remains green, but due to the accumulation of reddish oil, it becomes red in color. The zoospore lies liberated from the oogonium by the degeneration of oogonial wall. It rests on the mud at the bottom of the pond and remains dormant for the long period. Germination of zoospore. During the favorable condition, the zoospore germinate in the nucleus. The diploid zoospore nucleus undergoes zygotic meiosis to form 4 haploid daughter nuclei. The nuclei accumulate some cytoplasm to form 4 haploid daughter protoplasts. Each daughter protoplast develops a grown of flagella around itself and metamorphoses into a zoospore, often called a meiospore or meiospore. The meiospore are liberated by rupturing the zoospore wall. This slide shows zygote germinating and zoospore liberation. This is zygote germinating and zoospore liberation. After liberation, the microspore are the first surrounded by a delicate vesicle. The vesicle zone disappears and the meiospore get free into the water. After swimming for some times in the water, they settle on the some substratum. The meiospore withdraw the flagella and germinate into a new haploid Oedogonium filaments in some Macronandus dioecious species. Out of 4 meiospore, 2 develop into a male and the other 2 develop into a female. Under the certain condition, meiospore are formed insist of meiospore. In some cases, out of 4 nuclei, only 1 or 2 remains viable and the rest regenerate. This slide shows life cycle of Oedogonium. Thank you.